some people came to see me. One of them was Mike Christensen, and he was telling me that he'd observed that the endophyte has a rather unusual way of growing in ryegrass plants. One of the things that is a feature of fungi is that they grow by hyphal tip extension. So imagine a little tube like a straw with the tube extending at the tip, and it grows in between the plant cells in a ryegrass leaf and actually attaches itself to the cell walls of that plant. And what he noticed is that because ryegrass plants actually grow quite differently, they actually have a growth zone at the base of the leaf, and somehow the fungus has to actually grow through that zone to be able to infect an emerging leaf. But to do so, you'd imagine if it had hyphal tip growth, it would get torn apart as the plant cells were expanding. And so he had a theory or hypothesis that the plants or the fungus could grow. He changed its mode of growth from hyphal tip extension to something we call intercalary growth.
What we found out was that the growth of the fungus and the grass is synchronized. So when the leaf is growing, the fungus is growing. When the leaf stops growing, the fungus stops growing. The fungus is in place, and then it carries out its function, which is to do the chemistry, protective chemistry, that will protect the plant from insect pressures and also grazing animals. So what we proposed was that the fungus grows at the same place as the leaf is elongating, and that what would maybe happen is that the fungus attaches to the cells at the base of the leaf, and as those cells get bigger, the fungus gets stretched, and that stretching turns on all the processes that normally only happen at the very tip. And this this challenged a dogma that was probably over 150, 200 years old. And so we set about to show that this was true, and it was very hard to overthrow a dogma.
I've always been interested in microbial plant interactions, and so when the opportunity came to work with fungal endophytes, I grabbed that opportunity immediately because it's such an interesting system to work with. And so little is known about the molecular interactions between the host and the endophyte. And so consequently, yes, when Mike did come to see me about this new discovery, I was very keen to become involved because it did seem like a once-in-a-lifetime opportunity, and I think it actually has been. So since we got the work published, we were then able to get a Marsden grant together to look into the phenomenon in much greater detail. And over the last 3 years, we've had a PhD student, Samira Aryawansa, working on the project, and he's made tremendous progress in showing the mechanism that may be used by these fungi in order to sense that they need to undergo intercalary growth, because this mechanism is absolutely essential for the colonization process in plants. Without it, they are unable to get out of the crown of the plant right down at ground level and to colonize the leaf material. They have to undergo intercalary growth to perform that step. So the symbiosis is utterly dependent upon this step.